Hello, everybody. Welcome to Microbiology News on this channel, where we talk about some of the latest COVID-19 news and other related things to microbiology. I'm Faz Lam, and this is my co-host Danny Chan.
Hey, great to join. Yeah, this is our off week where we get to like kind of zoom out a little bit to find something that we want to zoom into, but also so we can talk about like the latest headlines that we saw and maybe the papers behind them. It's nice to do that.
Yeah, I mean, it's been kind of a slow week for coronavirus news, hasn't it?
Well, I mean, maybe like the specific like therapies and things, it's been slow, but there's certainly been lots to talk about.
Yes, so for those of you watching this from the far future, the President of the United States has unfortunately fallen ill from coronavirus.
Yeah. Oh, I'm sorry. I was gonna say like the way that I found out this news too, because I'm not a big news watcher. It was just like I was just browsing YouTube and it was like on the front, like every video in like the front section was like about this. I was like, oh gosh, like I guess this is big
Oh,
news.
oh man! Like I think the problem with me is like now every news source I have has suddenly turned into like all Trump hate all the time, and so it's been quite hard to get away from this. And I mean, I envy your like kind of ability to be like.
Oh, living under a rock? I don't know if it's the best
It seems
strategy,
like
but yeah.
I like to think of it as like a turtle, like hiding in the shell for safety. Like,
Yeah, I do feel like that sometimes, yeah. But like news ends up trickling, right? Like I'm just browsing YouTube to watch my daily videos and like there it is. I can't
yeah,
ignore it.
and yeah, I mean, obviously Trump news gets clicks and likes, so that's why we're talking about it. But from a
Yeah, we're no strangers to
yeah,
that.
but from the scientific,
But
yeah,
uh but
sorry.
oh no, I was gonna say, but like I think we're gonna highlight something different about it. We want to talk about the therapy that he's
Yeah,
receiving.
because as
Yeah.
president, he's going to get the best for therapy because what kind of country are you going to? You're not going to let him die, so you have to like give him the best therapy to make sure of it. I mean, that's so we're going to be trying to talk about some of that about so I think we can start by talking about Regeneron,
Yeah.
the com the
hmm
company that
it's a really big company. It's a really big
oh
pharmaceutical
yeah.
company.
I mean, a lot of the Pfizer papers that we reviewed have Regeneron as like co-hosts, because they're
Mm.
involved with the vaccine, they're involved with testing, they're involved with quite a lot of different things. So I mean,
hmm
we've probably encountered some of their papers before, just talking about the ongoing coronavirus things.
Sure.
And so I guess the thing that the treatment that Trump has been receiving has been the Reagan CoV-2 cocktail of antibodies.
Wait, what? Reagan? Oh, Reagan, like the
Regen.
first, yeah,
Oh, is it Reg?
like
Yeah,
Regeneron,
he's been having.
Regen.
It just yeah, it does sound like something out of Transformers. Just give me
Uh-huh.
the Regeneron for life.
Yeah, so a cocktail of antibodies. And those antibodies, this is the Baum paper, right?
Yep, the bound paper. So, uh, this is the paper titled "Antibody Cocktail to SARS-CoV-2 Spike Protein Prevents Rapid Mutational Escape Seen with Individual Antibodies." So,
Mm. Oh yeah, and for people at home, if you want to follow along, we have our Zotero reference manager, and we're dropping all the papers in there. So if people want to cross-reference afterwards, like what we're talking about, that's
yep,
that's
the
where it
links
is.
in the description as ever if you want to read along and to skip. Because we're not going to be able to cover every single paper we talk about this, but sometimes you might, if you see something you like, you might want to go into it in a bit more depth.
Yeah.
So this is yeah.
Yeah. So monoclonal antibodies, those are monoclonal, meaning like a single
Yeah.
clone, right? Like in our bodies, we get antibodies from all sorts of things, but modern science has allowed us to like extract the one cell that's making the one antibody and then assess how good it is. And then you can assemble a collection of antibodies from various monoclonals.
Yeah, it's like America's Got Talent, but for antibodies, where they try to pick out the best one, and
Yeah, the best
yeah,
one.
so and they pick out the best one, and once they have that best one, you just see it everywhere, and you can't get away from it, and that's the idea for the antibody, where because it's the best antibody, so if it's the best one for getting rid of the coronavirus, then that's what we want. We want to and for patients
Hmm.
who are infected, they might not already have those good antibodies. And I think we discussed before how the coronavirus antibody response can vary somewhat, and you don't always get the best antibodies in response. So having like this
Hmm.
parachute in and being like, we're the best antibodies, we're best at what we do, and oh gosh, I'm terrible
Yeah.
at explaining
So,
things.
but all these antibodies are for the spike protein,
Yeah,
though, right?
yeah,
Yeah. So like they want it's all antibodies against the spike, but I guess I mean in the title of this they're sort of suggesting that if they use multiple, then um, the spike can't like mutate to escape
yeah.
it.
So essentially, that's what they are looking at. So they treat the very cells infected with the coronavirus, and then they test to see whether any spikes can mutate away from them. And then I think they also have cocktail ones that are designed
Mm.
to stop those spike evolving, or looking at those.
Hmm.
So looking at areas that are mostly conserved, I guess. Um, I
Hmm.
didn't look too deeply into this, but um, so
Yeah. That's
yeah,
kind of cool. Yeah, cocktails and things. I mean, that's like the same principle as like any drug resistance, right? Like with like rapidly multiplying microorganisms, there's always that potential that they find a mutation that lets them still do what they normally do, but now without the pressure of the therapeutic that you've
yeah. I
deployed.
mean, the only they do they passage the viruses in cells and to identify potential escape mutants, which is a good
Hmm.
strategy, I guess. But again, I mean, I'm one of those people who says like if it's not in the real body, it's because I'm very skeptical of like cell culture things. So I mean,
Sure. Yeah, like maybe it never occurs to escape, right? Like maybe escape mutants don't happen that
yeah,
often.
maybe
Is
like
that
in
what
the body.
you're
Yeah.
thinking?
So, but again, it's a nice thing to have. It's nice to think that it makes it a more interesting paper that they thought about it rather than saying, "Oh, we've got an antibody that binds to something." I mean, lots of papers have that,
Sure.
but having
Sure.
something like this shows a little bit more forethought, shows a little more insight, which makes it a little bit more
Absolutely.
interesting to read those sorts of things.
Hmm. Yeah, we're also offering up for discussion this Hansen et al. Paper as sort of the maybe if people want to conceptualize how one would get
Yeah.
to a SARS-CoV-2 monoclonal. Um, yeah, this is like the a description of how that may come to be.
Yeah, so this one where because in the
Yeah.
first paper they just use cells and that makes fans go sad. So I thought I need to find something where they do it in animals and they take it into a humanized mouse model. So we talked about these to a fairly big extent. We've got like a 2 hour video on the humanized mouse models, and
Hmm.
I think we've covered this paper before in our like rundowns, but
I see. Yeah, this is the Regeneron one. This is like specifically about like just try to like talk about how it came to be,
yeah, so
right?
this is specifically about
The
how
actual
Regeneron came to be because since we've been keeping
or how regen whatever numbers come in it.
yeah,
Yeah, not the
whatever
company,
brand name they've
the
decided to
therapy.
call their antibody cocktail, because I mean they've taken it into humans and they say they've got good results. I mean, but I've only been
Hmm.
able to find press releases about that. So this is the latest hard like data I could be able to find.
Sure, the closest. Yeah, but like I guess right there. This is like actually I thought this was like a old like when was this published? This is published
21st of
like
August.
June. Oh
So
yeah, yeah. First release. Yeah, so it's been out there for a while. There are reports about monoclonals as early in our library under the interventions biologics section. Like we have some as early as May where people were talking about
yeah,
monoclonals, right? So this is like one of the other things that have been in the running for having use. And in some cases, these monoclonals are actually from
hmm.
convalescent people. So like these are antibodies that people have seen in people who recovered from it, right? You're kind of getting the biotechnologically produced version of someone's immunity, so someone's experienced immunity.
Yeah, I think not last week, but the week before, I think we talked about uh, a nanobody that was created from just a minor part of the. Yeah.
Oh yeah, that's right. Well, that's camels, right? That's like
Oh,
a
yeah.
really
We talked about
different
nanobodies, and then
antibody
they
in that
took
sense.
one where they chopped off just the very like tip of like the human antibody and used that as well.
Ah, yes, that's right. Yeah, they use like a section.
Yeah. So this is the kind of thing that we've been talking about, and but we haven't really covered it in much depth. And so, given that like this is probably quite going to be quite an important treatment going forward, because
Sure. Yeah, we could talk about one of these papers to talk more about monoclonals. And like biologics, it's a section under interventions because it's a big
yeah,
section of medicine, right? Like tocilizumab, we've talked about that one, right? Like, or at least like in passing, we've talked about it. Those are monoclonal antibodies, but instead of being raised against a viral protein, they're raised against some immune protein. And so they end up being like immunomodulatory, but same stuff, right? It's
yeah.
like making a bunch of antibody to
Yeah,
something
antibodies are
to
really fascinating like drugs. I mean, you've got like things like Herceptin, we've got a couple of other antibody drugs that are really effective. They're quite expensive at the moment,
Mhm.
but
Yes, they're very expensive. Yeah, because they're made in cell culture,
yeah,
right? I think that's one of the big limitations of thinking about widespread use of monoclonal therapeutics, right? Is that right now, like the types of drugs, I don't know, maybe Humira is one of the drugs that's made the most. It has like so many, like it's for psoriatic arthritis, like some autoimmune diseases, basically. But I mean, how many people in the world have an autoimmune disease versus how many people in the world might want this therapeutic? Like that, the scaling is a lot different,
yeah.
I think.
I think that's why they're talking about how difficult it would be to scale up and how they'd have to
Yeah.
like basically give it in the most severe of cases or the richest of cases, depending on
Oh yes, the wonderful thing about modern
yeah.
medicine.
Yeah, you still need that cash. We don't this stuff don't come for free.
Yeah. Okay, so let's see what else do we have on the
On
docket that we might be able to
Yeah,
talk about.
okay. Let's see. So, uh,
Reinfection and with SARS-CoV failure of humoral immunity, a case report.
yeah. Uh,
Oop,
this was one that you added, I guess. So, do you want to talk
Yeah,
about
I
this?
think I put this one in. Let me pull it up. Reinfection. Oh yeah, yeah. I mean, this is like right. I think this is that I'm obsessed with reinfection right now. I
Yeah,
think right because it's an interesting story. It's an interesting story to um, because we know about asymptomatic spread, right, and that should be informing this idea that people should behave right carefully, like wear your masks when you go outside, right. Try to not do too many activities where you come into contact with people because who knows, you could be a spreader unless you've been tested. Get tested frequently. And so, yeah, here's another one where we talk about a potential reinfection event. And I think this one sets up the story that I was seeing about a mutant, actually,
yeah.
D614G. It came up again in another paper.
I believe. Let me
Yeah.
see whether.
Is that the, oh no, that's spike mutation D614G alters SARS-CoV-2 fitness and neutralization susceptibility.
Yeah,
So there's like kind of two. There's a case report where they found this mutant, and then I think then there was a place where they did some studies to see like what does this mutant actually do, um,
yeah.
and presumably it changes some biological properties of the virus,
So,
which is different. You know, like this is the first news that we've had of this sort of stuff.
so I mean, we've heard about this a couple of times. This is like this because it did emerge during the pandemic. Because I know that I did hear news about like a vaccine candidate being created specifically for this version of
Mm,
the spike protein. I didn't put it in the
gotcha.
bucket because it was very much in the cell culture stage of things.
Sure.
Um, but this is an interesting story to keep an eye on. And
Yeah. I think definitely like because it does like if vaccines are being made for one versus the other, but this different form really does behave differently from the neutralization standpoint, then that would be actually a really big consideration for vaccines that are coming out.
I mean, the thing that because again, this goes into the failure of like humoral immunity, and so this
Mhm.
and this is
Meaning you could have antibodies, but they don't work, right? Like that's I think that's what this case study is trying to
right.
say. I mean, it's not a good, it's a case study, right? So it doesn't give us that great. It's only one instance of this happening, but like that's sort of the setup for then maybe the significance of the mutation.
Yeah, I mean that because yeah, case studies. I mean, also it does fit into. We did talk a couple of weeks ago about how B cell immunity gets screwed up in during uh, really severe COVID cases. So that's
Sure.
another thing to that this kind of might hook into as well.
Yeah, like I didn't look at what this paper was, but I assume that what it was is that there's like antibodies that are made that aren't good against the mutated form. But yeah, it could also just be that for other reasons, right? Like this humoral immunity raised against one form isn't applicable to the next, and that could tie into poor immune responses.
Yeah, it's something to look into.
Kind of cool. Have we looked at phylogeny yet in this?
We might have done. I think there was the MERS
short
that we looked at a little bit. The touch in.
Oh yeah, the MERS paper had it. Yeah, the one where they were passaging it in insect cell, or sorry, not insectivores had cells.
Yeah,
they definitely have phylogeny there.
We've touched, and I think there are a couple of papers that touch on it a little bit, but phylogeny is one of those difficult because it's quite difficult, especially with SARS-CoV-2, because there is this like.
Well, I mean, there's not that much variance, first of all, right, between all the strains, so that introduces an issue.
And there has been like conversation about different strains of this SARS-CoV-2 virus being out in the population, but there's been very contradictory information as to whether they have any changes in exert any changes.
Actually, I'm looking at figure 2, and we shouldn't be saying strains. I've heard this before. Right. It's like we should be saying isolates, because like strains implies there is some biological difference between them. Right. But all we know is that we isolated different versions. We don't know like if that's biologically relevant.
Okay.
Sorry if I introduced that word strains. It's not a good thing.
The more you know. Use the word isolate now. But yeah, no, that's I think that's a good point actually, because that's something that we're quite used to as microbiologists. So, like, we've got an isolate, it does something weird. Is it relevant? Usually, unfortunately, no. But if you can convince people, you can get a paper out of it.
For sure. Yeah, so we got some other things in this reinfection thing. We have asymptomatic reinfection of 2 healthcare workers from India with genetically distinct SARS-CoV-2.
Yeah.
That is that's very similar to like we had some news stuff, right, about asymptomatic or about reinfection, and here's another case study that's come through.
Yeah.
Once again, they find it through genetic similarity. So you don't know, now that people know that you can find it, like you can find it.
Yeah, and the more those data points build up, the more like interesting it gets, because then we can see, we can get more details about how this is working and why this happens.
But I think what's different is that the times that we talked about the infection before, it was actually 2 different infections, I think. Different severity of infection, but it was 2 different infections. Here we're having asymptomatic reinfection. So
yeah, that is interesting, because asymptomatic, you're assuming that they're asymptomatic because the immune system is doing something good. But if they can get reinfected, then that means that maybe the immune system, even if maybe something else has happened. Maybe there's like a low-level coronavirus infection that can happen that doesn't involve the adaptive immune system. It could be cleared up completely by the innate immune system, which is...
Yeah, for me, I think the implication is just going back to that whole like public life thing. Like, you got to be careful, right? Like going out into the public, because you could be spreading it. It's just like, you don't know, right? It's an argument for getting tested frequently, right? That sort of stuff. So, yeah. Asymptomatic. We're probably not going to cover that one. It doesn't look like there's a lot of figures in it. It's like 1 figure.
Yeah.
It's more like a report.
I mean, it seems to be like correspondence. So it's like kind of when someone publishes a result and then people want to go, yeah, I saw the same thing. And
yeah, exactly. So from that preprint, when people saw reinfection, now some more reports are rolling in. Other people can see it too.
Yeah. And I think the next one is recurrence of SARS-CoV-2 in recovered patients, which also is, so this is an interesting.
Yeah, it is kind of like I mean, they don't. Yeah, very related. Reposit. It's almost in the diagnostic line of things, though, right? Because they're talking about like RNA positive, so they're not like talking about any infection. They're just talking about like this is what this is a potential diagnostic pattern that we can see. How are we supposed to deal with it, right? Like this information is gonna inform like diagnostic procedures, I think. Because it says they still have to this indicates that it might not be. It's still important to affirm RNA testing and assessment of infectivity. They don't. Yeah, they don't necessarily know.
Yeah. So it's a weird result that comes up, and it's yeah. I can imagine this is the thing that makes people who are in the testing field quite paranoid because uh,
Mhm.
because it,
Well, I mean, it's just more of like when do you know like how many tests do you need before you know, right, that they're ready, people are ready to discharge kind of deal. And oh, it looks like they look into some like different ways of like different locations of testing, anal swabs or narrowfin. I didn't know you could get anal swabs for COVID-19.
I mean yeah because I mean they do like sewage testing for COVID-19 as well
Right,
as
right,
that
right.
has yielded some kind of association with coronavirus outbreaks. Um,
Mhm.
but I mean, there is that idea that different mucosal sections can be linked. So sometimes you can get a virus that can infect your nose in infecting your gut. I mean, there was a story I
Yeah.
read about how I mean about how Google searches for stomach bugs and kind of stomach flu symptoms preceded like coronavirus outbreaks. I I
Sure.
didn't talk about this, Joe, because I don't really believe that, but it's
Oh, like just like if it was like a symptom or something,
yeah,
right? This is like people that are trying to infer epidemiologic stuff from like social network information,
uh,
right? It's like just search results. Yeah.
yeah. I mean, my skepticism is that okay, well, restaurants are reopening as well. Could be that the people are eating out and getting, and
Sure.
getting stomach bug from
Hmm.
having eating at McDonald's. They're like, I mean, it's been
Yeah.
like 6 months, over 6 months since I've had McDonald's. I don't think my stomach could take it anymore. I think the lining's gone. So uh, if I eat a
Yeah.
KFC, I might die. I don't know.
The special adaptation that comes with eating fast food.
I,
It's just like
I
worn
feel
off.
like we're losing that, com- and we're the moment I
And,
walk into like a KFC and order that double down and then just drop.
yeah, obliterates your GI tract. Yeah, probably for the best. You don't want to be too adapted to that type of food.
Yeah, but I mean but this is sorry. Going back to this paper, I mean it's interesting, but also like, in terms of what the significance of negative tests as well. So being
Yeah,
discharged for a negative,
and we haven't talked a lot about testing.
yeah,
Yeah, and what it means to be tested. Yeah, it might be an interesting one actually. Now that I think about it, it's the very different data than what we're normally looking at. It's small though.
yeah.
It's only just like yeah, a few tables.
I mean it's interesting because I remember like I was doing when I was looking at flesh-eating bacteria, I did nasal swabbing with mice, and sometimes mice would be who were infected, like definitely infected with would suddenly turn up blank on tests and then later come back with like
Sure.
so
Yeah, I mean that again, like it could be. It's like right. There's error in
yeah.
sampling techniques.
So error in, I mean the limit of detection can be might be an issue where so
Right.
it's
And then again, like this is why this paper is being so careful to talk about like like that it's just RNA positivity, right? Because like what does that correlate to actual infectivity? Question mark,
yeah,
right? That's for another paper to talk about.
yeah. Um, okay, so the next one
Okay.
we're looking at type 1 interferon susceptibility distinguishes SARS-CoV-2 uh,
Mhm.
from SARS-CoV. Now, oh, this is an interesting one because it brings a little some of that history back.
Yeah, yeah. Let's see, where does this data come from? I mean, I was interested in it because, just any differences are nice to know, right? Differences that we can see. And I feel like they would probably have a strong base of data maybe from samples that they got from classic SARS. But where does this data actually come from? Uh, the abstract. They're like novel COVID. Oh, they're doing it in cell culture.
Yeah,
So, yeah. So whether people respond like this, we're not really sure. It's a cell culture thing.
yeah, and
Oh, but they are talking, but they are trying to link it to or different open reading frames that exist in these two viruses.
that's interesting. You're looking at that.
So this actually goes back to maybe some basic information, right, that we could pick out about the other proteins. What are the other proteins in SARS doing?
Yeah, I mean, seeing because this ties into I mean, yeah, because I mean it's difficult because like trying to extrapolate from cell culture, it it's quite difficult. It is
Well,
possible, but
that's what I'm saying. I don't think this is about. I don't think we should be thinking about this as like something relevant for people. This is another. We're back to the like things end up being a tool to learn more about like the virus itself, right? So this is the phenotype in cell culture that can differentiate these two viruses, and through that they can identify what's the genetic factor in this virus, right? That might be driving this change, right? So learning more about like that accessory protein, whatever it is.
yeah, yeah,
Yeah.
that's right. Because
What is that? What does that ORF do? How is this virus like circumventing the immune system? Whether or not like there's an actual difference in the interferon, because like there's many ways to stimulate interferon. Maybe these cell culture systems, right, are lacking some of those ways of doing it.
yeah, yeah. Okay, so let's see.
It's a very simple paper. This is like it's like a really nice one if we just want to talk about like a study that's done in cell culture with infecting cell cultures.
Yeah,
It's like some
it's nice and straightforward. I like nice and straightforward papers.
yeah.
Next one is furin cleavage site of SARS-CoV-2 spike protein is a key determinant for transmission due to enhanced replication in airway cells. So, furin cleavage is something that we've touched on a little bit. We did a study on TMPRSS2 cleaving the SARS-CoV spike protein in order to get to fuse. This is another cleavage site that they're talking about here, and they discuss it a bit, but then they also look at the replication in airway cells, and they use a ferret model as well for looking at the effect of transmission. And that linkage between something that happens on a molecular level to something very macro like transmission is quite interesting to me. The idea that this one gene can actually have an effect on like how much air droplets a ferret can create. Yeah, I mean, I'm going to say will affect humans in the same way because the ferrets here are like an influenza model that they've adapted to SARS-CoV-2. So I don't quite know exactly what whether they have that ACE2 like or whether they have the similar infection and.
Yeah.
And also,
So wait, it's not droplets that change. It's just like the amount of virus that they shed, right?
yeah.
Changed. The ferrets shed less virus.
So they basically test it in a like
co-housing ferrets.
So they house like infected ferrets with uninfected ferrets to see whether they spread. And they found that when they got rid of this particular mutation, it spread less. So I mean, I think that this could be an interesting one because again I've been wanting it ties into the life cycle a little bit and it ties a little bit on the macro level.
Yeah.
I mean, I just realised that this actually took place at my alma mater, so I actually have been around the ferret lab taking place. But
you've seen the facilities.
Yeah, I mean, it was like being in a spaceship. I think I saw them when they were being opened up, like so.
Gotcha.
Yeah, I mean, the technicians are legally required to play with ferrets for 2 hours a day, apparently, because otherwise, because like all predator animals, they're smart, so you need to play with them, otherwise they'll get bored. So, because I do remember like that was quite fun. This had little tubes everywhere for the ferrets to crawl into.
Oh yeah, like because they like I guess they like exploring.
Yeah, even the ferret cages had like these tubes in them that they could like go into like different areas. So it was like super high tech, and the ferrets are very cute. So
yeah, I mean we I don't think we've talked about the ferret model. But yeah, it's a influenza model for sure. We know that they can be infected. I think there was like a paper that talked about like all the different animals that get infected by this. But yeah, we talked about the a hamster model, or no, Syrian hamster, golden hamster transmission model. Yeah, this is interesting. I mean, this is nice because it does like blend together. This is sort of like one of those papers that can only exist now that we've seen a lot of basic information, right, that has existed. Because it's like very much like we have to know the furin cleavage site, right? That's been identified before, right? You have to have like a model of transmission and ways of talking about that. Yeah, interesting.
Yeah.
What else do we have in here? Enzymatic activity of the NSP 14 exoribonuclease is critical for replication of MERS-CoV-2 and SARS-CoV-2, or MERS-CoV and SARS-CoV-2.
Okay, this seems like quite a meaty paper. Because non-structural proteins are things that we don't hear much about because they don't end up in vaccines or other things, but they're quite important to the life cycle of SARS-CoV-2 and other coronaviruses apparently. So, and
yeah, I think I'm once again on the hunt for finding out new things. There's a lot of spike talk and stuff like that. Let's talk about some other proteins that might exist and what they're doing in the infection. So yeah, looks like this is also cell culture.
Yeah, but I mean, that's not so bad because again, we are looking at the virus. So,
exactly.
So
we're studying the virus more than we're studying. Like we just need like some sort of infection model to see what the proteins are doing.
I mean, it's not like they're adding, I don't know, some seaweed extract and going, okay, well, now we can cure it with seaweed. I mean, they're actually using it to try and understand about how the virus works.
Yeah. It's interesting. I mean, I just haven't heard about this particular protein that's being made. I think that's always, it's just fun to learn something like that. A lot of biochemistry, it seems. Lots of biochemistry.
I mean, talking about like mutation frequency and a lot of the kind of nitty gritty of how to do RNA-dependent RNA polymerization it's also a unique kind of viral metabolic kind of process that
that we don't see in other. Yeah, it's like because where else do you need to make RNA from RNA, right?
Yeah, I mean the whole basis of a lot of cell immunity is the fact that RNA-dependent RNA is not allowed; that it's like forbidden. I mean that's why last week we had all those pictures of lipid balls filled with RNA.
That's right. Yeah, exactly. That's like it's a very alien biochemical process to be happening inside of a cell.
Yeah,
and so.
Yeah, man, that was a fun paper last week.
It is cool. Yeah, it's like great. I love those pictures that we saw.
Yeah. This is.
This is a very old school biochemistry paper. Like they're doing gels where they're looking at the actual activity of cutting up strands of RNA.
Oh, that is old school. Like restriction site like mapping. That that is classic. Where you just take a length of DNA and you want to find out where your gene is, so you just cut up the pieces and try and figure out which piece it's on. Is that right? Or
no, it's like I guess like they have because like this has some biochemical function, right? In vitro exo-N activity. So they have like strands of RNA and they incubate it with this protein and they see that it gets chopped up in certain ways.
Okay.
Right?
Oh, that's more that. Oh, right.
RNA is the substrate of this NSP14, or that's what they're trying to show.
Right. Okay. Silly me. So they're actually trying. So this is some sort of pro-proofreading activity or something like that, and so they're trying. But yeah. So.
Okay, so that's like an old school biochemistry option that we have.
Yep. Next one is
another NSP.
Yeah. So we talked about this. Oh no, we didn't. Structural. So NSP1. Structural basis for translational shutdown and immune evasion by the NSP1 protein of SARS-CoV-2.
This one I think maybe we have even more. I mean, this is a published. Oh no, this is still. It looks published. It's not. It's still. It is published.
Because I got the bioarchive link for some reason, but I ended up. But now there's a little thing that says like now published in Science, and it's got the Science DOI. So
because this layout looks like it's a nice layout. So I was like, this sounds like a
bioarchive.
But this is really going into the mechanism of this immune evasion protein, how it gets into, what is it binding to? A ribosome.
Yeah. So yeah, obstructs entry into the mRNA, the tunnel on the ribosome. It
Hmm.
so it can block the innate immune responses in the cell that detect double-stranded RNA, I guess, or the thing that Rig1, if I remember correctly, is well, it's written there, but I'm trying to remember what Rig1 does. Um,
It definitely binds
yeah,
RNA. Right? It's one of the TLRs.
yeah, that's right. So
Intracellular TLR, just like floating around in there.
So tolerant receptors are these innate immune-like kind of proteins that spot things that are generally deemed to be foreign and grab them, and then based on that, they can get cells to apoptose. They can do a response that seems very much like an antibodies. This is the complement system and TLR system are very like evolutionary old, and yeah. So
And they're using cryo-EM to do all this structural stuff. So we're revisiting our favorite technique. But this is just to see the molecules. So they're not looking at big structures. I mean, they're big molecules, I think, but yeah. Oh, weird. So it's like they're talking about it as it suppresses translation, right? It's like, oh, you want to make the proteins that like are going to shut us down? Like, no, you can't make proteins.
yeah.
I'm actually curious. That's a weird one. Like, it's a weird mechanism to be able, because you need the virus also needs the ribosome to make its own protein. So why would blocking translation be the way that it shuts down the interferon response?
Oh, you're right. I think this article's clickbaiting us. It's like
yeah.
got us.
Well, I mean, it's like. This is I'm interested. Yeah, it's true. It's like make me interested in the story. Like, why would that mechanism do? The science articles rely on this type of clickbait as well, right?
Yeah,
like they need people to read their articles.
Yeah, they want people to read and cite their articles, and hopefully,
that's
what all journals want essentially. So
right. I mean, that's been a criticism I think levied at some of the. Like the cell nature science, or more like the nature science, right? Editorial outlook, right? Like they want to publish things that are like groundbreaking, but later on, like they might be kind of reinterpreted as like, oh, actually that was a mistake.
Oh yeah, it's tough because trying to find what is going to be a good like article is very you never know, and and sometimes like you can't necessarily. I mean, for publishers, they generally aren't experts in whatever field they're trying to do published work in. I mean, sometimes they are,
for sure.
Then they can get bamboozled. So exciting stuff.
And I mean, I think the other thing is, like if you make a commitment to publishing like only the most novel and exciting things, quote unquote, right? You're gonna get things that are like, right, that might not be fully fleshed out later on. Like as people find more information about them, it recontextualizes them in maybe a more mundane circumstance.
Yeah, I mean, they get what will get people read. I mean, all journals survive on getting people to read them, but science and those big ones especially, they're very aware of the eyes that are on them, so they do try, but occasionally things get through. It's just human. The fact that this is all scientific endeavor is run by humans, and we do mess up a lot. So
for sure.
I mean, things like bioarchive help because that means that people can pipe in the comment section and point out flaws immediately. So let's see. Yeah, no one's popped up. Sometimes you see some fun things in the comment sections, but.
Okay, and then next we have another lab model that we might be able to talk about. Mouse-adapted SARS-CoV-2 induces acute lung injury and mortality in standard laboratory mice.
Right.
So I guess we're like really looking at like again like why the mouse is dying, and hoping that that's like close to the way that humans go down.
Yeah, because trying to figure out like why people die, because again we don't get that. Because with mice, if we can get something similar, we can probably get a lot more insights about humans. Because there are a lot of things we can do with mice that would be ethically unjust for humans, perhaps.
Sure.
Legally unjust for humans, and for things that would get you sued. But if mice had lawyers, would be screwed. I
mean, that's kind of like when you get too close to animal rights groups, right? Like that's the danger.
Yeah, I mean, So I mean image rights alone would be screwed. But
Mm-hmm.
yeah. So and we talked about like
Oh.
host like I think have we talked about this mouse adapted SARS-CoV-2 virus before, or is this?
Maybe I think this is a different mouse adapted SARS-CoV-2 virus potentially.
Right.
We definitely did talk about a mouse adapted SARS-CoV-2. That was because in that one it was like it was sold as a good research tool because your favorite mouse model. Oh no, that was different. That was a virus that made the mouse have the ACE2 receptor. I don't think we talked about in depth
I think we
the
talked about
opposite
both of them
version.
in that double header one.
Oh, maybe we did. Yeah. But yeah,
I
this is
think this
one
is
of the ways. But I'm not sure if this is the same
actually, I
virus.
think it is because it's SARS-CoV-2 MA. I think that was the one we worked on.
MA10.
MA10. I mean, yeah. Well, yeah, they did passage it a couple of times, and they've been putting into different. Here they put it into different strains as well.
Mhm.
Because again, I think that's something they did in the other one because just to demonstrate the advantage over because this one is good to go on lots of different ones. So
Yeah.
different strains of mice, which do actually behave differently depending on certain conditions. So I am allowed to call them strains, right?
Yes, absolutely, absolutely. Well, they called theirs MASCP6.
Oh god,
I,
mouse adapted
I
SARS
don't.
coronavirus passage 6.
Yeah, passage 6. So I don't know if this is the same as this M10 one they're talking about
Yeah, it might be.
here.
Um,
Yeah.
but
I mean, yeah, exactly. The concept is the same. Keep giving it to mice until they end up getting sick from it.
yeah. So the question is, okay, fine, but what are we learning from this? Is this just justifiable? And they are
Exactly.
looking a lot into like the immune response and about and the symptoms and so I so I'm trying to pick out the figures for this.
Yeah, we didn't put it in here as a paper. I thought I did, but I guess it's just.
Oh no, it's fine. I mean so they do lung function, which lung function research in mice is very complicated. Because I mean, I remember like one of the first lectures I had was on this thing called PenH, which is measured in, which is this statistical like thing number that's derived from a number of different factors that my lecturers decided to teach me, and then say, "Oh, you should forget all this because PenH is irrelevant." And I'm like,
Oh my goodness. So it's a metric of.
it's a metric of lung resistance, I think. And
I see.
and I'm not sure that my professors were entirely right in writing off completely because I've seen it pop up again time and time again. So it's just one of the things that I think it works when you understand how it works and what the underlying factors are. But
Sure.
that in itself is the barrier because even though I taught this in lectures, I do not remember what. But
Sure.
but yeah, lung function is it? I mean, because measuring lung function in mice can because it's hard to hook mice up to machines small enough to measure resistance or even. So I remember we had these like big like kind of special cages with like which measured changes in pressure at a. So it's tough. So but that's an interesting way to approach it.
Yeah, it's cool. This is it's a big paper. It's chunky.
Yeah, it is very meaty.
Yeah.
Let's see.
So yeah, this is like mouse model and like the challenge of doing like a tissue damage, I guess, version, right? Like it's more specific than just the mice replicate this, and now we're looking at it's like somehow affecting this metric, these lung function metrics.
And that's something that's quite important for human infections. That's something that we can monitor in humans because
Right.
I think that does make it interesting in that sort of. But yeah, because again, like ARDS gets bandied around a lot in mice, but actually the ways you measure it can be quite difficult and quite specific. And sometimes we like say ARDS based off of lung histology, and oh look, we've got some things that are characteristic of that, but we don't actually see how it occurs and develops as the mouse is alive.
Sure.
Whereas this kind of gives that
yeah.
opportunity to look at
Because this you could see like, oh, the breathing's getting more shallow or something over time, right? You get more of that information instead of having to sack and say histology. This is broken.
It gives you another metric to say like whether your vaccine is working, whether your treatment's working, because it's nice to have an outcome that isn't the death of the animal, because that allows you to get a lot more information that of like the potential for recovery and the potential for like
for sure.
Yeah.
Okay, so that's our animal model one. Oh, and then we have this. We mentioned this before, that there's this spike mutation, D614G, alters SARS-CoV-2 fitness and neutralization susceptibility.
Yes.
So this is like the in-lab follow-up to the report, right? That reinfection with this variant, and then here's like some lab studies to say that, hey, this variant it does have some biological differences when you put it inside of a model. What models are they using for this one?
Yeah, they're using hamsters.
Hamsters.
Let's discuss those. Those are good models for coronavirus. Because they got humanized too, so they should have a human-ish infection.
Well, more closer related ACE2 to us.
Yeah, one that the SARS can bind to, at least I hope.
One that the SARS can bind to.
So yeah, they compare the D614 and the G614 in hamsters, and they find that there are some differences to the infection, and that's important for human infection as well, because I think I think population level data has been contradictory so far for D614G, but it's not something I've really delved into that much. So yeah. But I think it could be important, because most of the way I've looked at it is through vaccine studies where most of them say, oh, our vaccine will neutralize this as well. That obviously loses a lot of like very important information that about how the pathogenesis changes. So again, it demonstrates how much you'll get limited if you only think of vaccines alone. There's all this other pathogenesis and life cycle that really informs and is important to understand how coronavirus works.
For sure. I like this paper. This is like a lot of heft to it. There's also an image of the Syrian hamster in extended figure 1.
Oh yeah, I'll bring that up quickly, but yeah, I did see that, and I was wondering like, oh my.
That's what they're sacrificing over and over again for us.
I don't know why researchers try to make the animals look as cute as possible. Maybe this like
just a reminder.
Just hey, by the way, we're monsters.
Noble monsters.
This is what. Yeah. Oh gosh. And yeah, they're showing like the hamster. Is that the hamster lung?
Or a picture, the cartoon of the hamster.
And also pictures of like adorable hamsters, and But I think it's important to understand that this is a sacrifice that people are going to try and cure people of coronavirus. So
yeah, they also do I don't know if we've talked about this sort of model though. They do replication in primary human airway tissues.
Okay.
So they remove human airway tissues, I think, and put them into a cell culture sort of condition, ex vivo work.
I know that's something that we try doing. We did that with tonsils, but it's something that is becoming more important, I think, to confirm that what you see in animals works in humans.
Yeah, it's a nice bridge, right, for the animal model studies to add some of that clinical the human relevance, the relevance to humans.
Primary cells are good because they are from live people. The difficulty is actually getting them, because you have to find someone who's willing or in a situation that they can provide that. And you know, I need my lung cells, so I don't really want to give them away. I mean, I'm using them right now.
Yeah.
That's
okay. And then we got some vaccine stuff on the docket.
Yes, we got some vaccine stuff. Johnson & Johnson have come out with their adenovirus 26 vaccine. Again, it's a phase 1/2 trial. So it's a phase 1 trial, but with added like antibody and T cell results. And they basically seem to find that it seems to look okay. It
Hmm.
doesn't look partic. I mean, the primate studies were really good, but this seems like about average for the safety compared to the other vaccines that we have at this stage. Um,
Hmm.
but it's good to know that they've made it like into humans, and there aren't any like massive side effects, which is always good. But
Hmm.
yeah, again, this is paper. If you're interested, you can
Yeah.
download and read this. But it is very much very similar to other papers of this sort.
Yeah. Yeah, we're sort of, I think we said this last time too, like at some level, the clinical studies papers, they're very similar to talk about them, right?
Yeah,
Like we're seeing the same stuff over and
they do
over.
tend to blur together if you've
Um,
seen enough of them. It's like a lot of them can use
right.
the same.
But I'd be really excited to read one when they have like at least something like maybe interim phase three
Yes.
stuff because I'm wondering like they'll probably look at neutralizing antibodies, but they're also going to look at like just infection rates, right? And like talking about that idea of like immunizing a bunch of people and then waiting for like monitoring for people's infection. Like that's like an interesting thing methodology that we haven't been able to talk about.
Yeah, and also they are talking about challenge studies in the UK. Something that
Hmm.
I that just always worries me when people talk about that. I can't because I can't help but think
Yeah,
like
it's like I heard that in the UK, there's like this it's like where healthy volunteers, right, can volunteer their bodies to enter these challenge studies.
yeah, I just think
There's like
yeah.
some nonprofits that are trying to collect the volunteers for
Oh
it.
yeah, that's been going on since like the beginning of the outbreak. Lots of people who are willing to like sacrifice themselves for the good of the world, and I can understand the intention behind that, but I do also like remember the cases where people have been through these studies. So like gene therapy was like held back by nearly a decade because people ran a study like where they gave people gene therapy and someone died,
Mhm.
and then that held up because of
Yeah,
that. Every single study after that was held up. So I'm very worried about that, about doing that sort of thing
sure,
because yeah,
sure.
although now with antibodies like Regeneron, new all these other treatments for coronavirus have started to make challenge studies a lot more feasible. So
Yeah, it is interesting, right? Because then there's a therapeutic, like if you have enough of that therapeutic ready, then that's the right.
yeah,
People have a clear like clinical path that they can take if the infection takes a turn for the worse.
yeah. So there is that. Um,
Yeah.
you also had like data on the failed Pfizer vaccine candidate. So BNT162b1 is a vaccine candidate they chose not to go with, but they have published a study called COVID-19 vaccine BNT162b1 elicits human antibody and Th1's T cell responses. Uh, so this is again focusing on the T cell
Hmm.
response, which is quite an important part of the immune response, and we want to see that when we give an a vaccine. So yeah,
Hmm.
this is uh, fairly good. I mean, but yeah, it is almost like an addendum to the other phase one trial that I think we discussed before
Sure.
for Pfizer. And
Yeah, we had we had discussed it in its preprint form,
yes,
right? And then when they published the BNTB2 version, yeah, then we talked about it a little bit
yeah.
again.
So this is interesting from the aspect of like vaccinology and how it triggers that innate immune response. Uh, but
Hmm.
again, this is with the vaccine that they've chosen not to go with. So
Yeah.
yeah,
So not that in maybe not yeah.
uh, and then we have Moderna, who have come out with a safety and immunogenicity of SARS-CoV-2 mRNA-1273 vaccine in older adults. So this is focusing on the main at-risk group for SARS-CoV-2. So this is
Yeah.
somewhat interesting for the fact that the older population are the one we're most worried about. They're more likely to catch SARS-CoV-2. They're more likely to have symptoms,
Yeah.
and we have to make sure that they, and also in terms of immunity, like the immune system,
Yeah, their immune system works a bit differently.
yeah,
As with everything and with age, it changes and degrades.
yeah. So trying to see whether this works or not, um, and it does seem to generate neutralizing antibody titers. So
So this is just like this is still like phase one, phase two. They just like expand like are focusing on like a subset
yeah,
of like older patients that they've enrolled.
yeah.
Hmm.
So I think each of these studies do like kind of section off by age group. So the phase one studies had like different age groups and different doses, and I think this one is where they
Hmm.
segmented it off. But it's good to hear something from Moderna. It's been a while.
Sure.
Yeah. So that basically covers it on the
Okay.
vaccine end of things.
Yeah. So I don't know if we'll, I don't think we're going to talk about a vaccine paper next
No.
week. Like these ones too, they're very much like updates to stories that we've already seen.
Yeah. But obviously it's
Yeah.
making the viewers aware of these because I know that people are just like waiting for a vaccine and so this is
Yeah.
something that if people are interested, they should definitely check these papers out. Um,
Hmm.
yeah.
Hmm.
What else?
So we could talk about the antibody cocktail, right? Because we haven't talked about monoclonals that
Yeah.
much.
I'm leaning heavily
Uh,
towards that because Regeneron is it's just a great name. I love that. And and it could be responsible for saving the president's life. I mean, that could be a good title for a YouTube video.
okay,
The
very,
the cocktail that saved the president's life. I mean, I'm not going to say Trump because I know
that's
that'll turn people off, but president people that's a neutral term. People love that.
um, and then I guess we could be doing. Oh, we could be doing the D614G alters SARS-CoV-2 fitness and neutralization susceptibility. That's the hamster,
Yes.
the infection with the hamster, and then we could do any of the NSP ones, I would say.
Yeah. So yeah. Uh,
Yeah, well, let's not. We're not going to do the case report.
no.
Both case reports. Oh yeah, I guess even the diagnostics one, maybe not.
Yeah. I'm leaning against the acute lung injury one as well, um, because I don't want to have to relearn those old lectures on resistance and everything.
Sure,
um.
sure.
Uh.
Yeah, I wasn't so excited about that one either. I mean, it's a mouse model, which is interesting, but yeah, when looking at that paper, it's like, okay, it's very technical in that way, right? Of finding a new measurement.
Yes. Um. Okay.
Yeah, I'd be down to talk about which antibody one would be the
Yeah.
best one to
That's
talk about.
so. I mean,
How,
I'm leaning.
how the antibody cocktail was generated.
Yeah, I'm leaning towards the mice, but the antibody cocktail, how it's generated, could also be quite interesting. I think in terms of just understanding the logic of how these are created. Um,
Hmm.
so
So that's oh, you're leaning towards the Hansen one because it's actual mouse
yeah,
data.
I'm leaning towards the Hansen one, but I can understand. But I'm so divided on this because I can understand exactly why you don't want to, why the antibody cocktail one would be good as well. Uh,
Yeah. Ooh,
hmm,
interesting. Yeah, mouse data. Oh, it's like it's the difference between knowing how it's made versus how it might work.
yes. So
Yeah, because like the one where it works complex, there's only four figures in it. Well, and then there's like seven extended figures.
yeah, I mean, science papers, they always like, they kind of deceive you a little bit, going, oh, there's not much in there, it's only 2 pages. And then, like, everything else
Yeah.
supplementary is like 20 pages.
Right.
It's point you don't even need to read the original paper, just download the supplementary and everything's in there. But,
Right. But there's not that much in the how it's made one is actually small. I don't see that much. Or I mean, I don't have the supplement in front of me, but figure S1, I don't see the figure S's going up that
okay.
much. We could both. We could do both.
I, I think we'll pick one maybe. uh,
Okay.
yeah. Um, how, so I think the,
So actually, the meteor one is the mouse one,
yeah.
I think.
Uh,
Yeah. So let's do Hansen.
yeah, okay, we'll do Hansen. Yep, studies in humanized
Hmm.
mice and convalescent humans yield a SARS-CoV-2 antibody cocktail.
Hmm.
The cocktail that'll save the president's life. Is this cocktail bad enough to save the president? Oh,
Cool.
yeah, cool.
Sounds good. Yeah, so if you guys are reading along with us, then make sure to tweet us any questions you might have using the hashtag. Yeah, and next week we'll be back and we'll dive into this figure by figure and figure out how good this antibody cocktail is. Expensive antibody cocktail, probably.
Oh yeah, expensive,
extremely,
but
extremely pricey therapeutic.
but well, what price can you put on a human life who is rich?
Yeah, 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 right, right.
Uh, yeah, definitely. So
All right.
yeah, thank you for joining me. This has been great. Um, yeah, we'll see you next week.
Mm-hmm.
Yeah,
See you next
see you
time.
next time.